on the warm scrolling trace of an EEG. A newborn brain is already at work. The infant lies still. The room is dim and close, the air held at a warmth that barely differs from the temperature inside the womb it left only days ago. A few fine wires run from adhesive patches on the small, curved skull, translating the electrical life of the brain into a moving line on a screen nearby, a line that rises and falls and rises again, scribing patterns so ordered, so rhythmically precise, that the researchers who first described them were struck not by their strangeness, but by their purposefulness. This brain, not yet capable of memory or language or the recognition of a face, is already cycling, already oscillating between two distinct electrical states with the regularity of a tide, completing a full revolution through both states in roughly 50 minutes, over and again, across the whole of the night. The line lifts into low, mixed frequencies. It is a restless, irregular trace, the brain flickering with something near wakefulness, the eyes moving behind the closed lids, the small muscles of the face and hands producing their brief, involuntary twitches. Then, after some 20 or 25 minutes, the trace begins to slow, the amplitude rises, the waves grow wide and sweeping, rolling across the screen like the slow passage of something very large and unhurried. And then the whole cycle begins again, the quick return to the rapid, mixed frequency state, the gentle subsidence back into the slow, repeating until the morning comes, or the hunger comes, or the sound of something close interrupts the rhythm and pulls the small sleeper briefly back to the surface. This is the ultradian cycle. The word means simply shorter than a day, a rhythm that recurs not once every 24 hours, but many times across a single night. In a newborn brain, it completes in about 50 minutes. In an adult brain, that same oscillation has stretched through some combination of developmental pressure and neural maturation to somewhere between 90 and 110 minutes. The underlying rhythm is the same in both cases, the same alternation between two broad sleep states, the same tide of neural electrical activity, pulling inward and then releasing. What changes across the years of growth and the decades beyond is the period of the rhythm, the time it takes the tide to come and go. Sleep is not a single thing. It is not a switch that flips, removing consciousness and restoring it some hours later, unchanged. It is a structured sequence of states, each one physiologically distinct, each one doing something different to the brain and body, and that structure is not fixed across a life. It bends, it shifts, it deepens, and then slowly, over decades, retreats. The architecture of a night's sleep at 5 years old is almost unrecognizable compared to the architecture of a night at 75, even though both are called by the same word, sleep. The adult version of this architecture moves through stages that researchers have mapped with some precision. The process begins gently, in the lightest layer of NREM sleep, a stage where the brain is quieting, but not yet committed to depth, where the body is relaxing, where the boundary between sleep and wakefulness is still permeable enough that a soft sound might bring the sleeper back. From there, sleep deepens into a second NREM stage, where the EEG begins to show two particularly characteristic features: brief bursts of electrical activity, lasting less than 2 seconds each, and a particular slow oscillation that marks the brain's growing commitment to the night. Deeper still lies the stage that researchers once called slow-wave sleep, the territory of the largest, slowest electrical waves measurable in the sleeping human brain, waves moving at fewer than 4 cycles per second. Waves so large and so low that on an EEG trace they appear as the broad valleys of some slow geological feature. the kind of rhythm that seems to carry its own weight. And finally, rising out of this depth, the brain arrives at REM sleep, rapid eye movement sleep, the stage associated with the most vivid dreaming, characterized by a brain that becomes nearly as active as it is during waking, while the body's major muscles are held in a state of near paralysis, disconnected from whatever the dreaming mind is doing. In a young adult, this sequence repeats 4 to 6 times across a full night's sleep. The early cycles are weighted heavily toward the deeper NREM stages, toward slow wave sleep, toward the restorative electrical depths. The later cycles, in the small hours of the morning, shift their balance toward REM. The NREM stages grow shallower, the REM periods grow longer, the night's dreaming concentrated in the hours just before waking. The whole arc, viewed from above, looks like a tide that comes in deeply at first, then retreats, then comes in again a little less deeply, the waves of depth growing shallower with each cycle, until the tide goes out for good with the morning. A newborn's EEG knows nothing of this architecture. Not yet. The categories themselves, the defined stages of NREM and REM, do not quite apply to the immature brain, because the electrical signatures that distinguish one stage from another have not yet fully differentiated from each other. Researchers who study infant sleep use different terms: active sleep and quiet sleep. Active sleep is the state that will eventually become REM. Quiet sleep is the precursor of NREM, but neither is the finished form. The brain has not yet built the circuits that will generate the specific oscillations defining each stage, and so the EEG trace of a sleeping newborn is not a miniature version of an adult sleep trace. It is a rougher, faster, less differentiated signal, the sketch from which a painting will eventually be made. 2 features of that future painting are worth sitting with for a while, because they shape the entire lifespan arc. The first is slow wave sleep, that deep low-frequency territory, measured by the power of delta oscillations on an EEG, oscillations between half a cycle per second and 4 cycles per second. The second is the sleep spindle. The spindle is a small, precise thing. On an EEG recording, it appears as a brief waxing and waning burst of faster electrical activity, rhythms between 12 and 15 cycles per second, generated by a loop of neural signaling that runs between the thalamus and the cortex, a circuit that pulls those 2 structures into a brief synchronized oscillation lasting less than 2 seconds. On the EEG trace, it looks exactly like its name, a narrow, symmetrical shape, widening slightly in the middle and tapering at each end, appearing and disappearing in the otherwise relatively quiet terrain of NREM stage. N2 sleep. It is a delicate structure, the spindle, precise, nested within the broader architecture of the night, repeating several times each minute during NREM sleep in a healthy young adult brain. Spindles are absent at birth. The thalamocortical circuits that generate them are present in their basic form, but the connectivity, the myelination, the fine calibration that allows the loop to resonate at its characteristic frequency, these come later. The first spindles appear somewhere around 4 to 8 weeks of postnatal life, as the relevant circuits begin to consolidate and fire together. From that point, spindle density, amplitude, and duration grow across the childhood years, reaching something close to their adult form during the school-age period, and then beginning a slow, decades-long decline from somewhere around the 4th decade of life onward. They grow more sparse, shorter, lower in amplitude. The circuit that generates them does not fail suddenly. It quiets gradually, over years, its output diminishing so slowly that no single night marks a change. The story of slow-wave sleep is similar in shape, but even more dramatic in its arc. Those large, sweeping delta oscillations, the ones that mark the deepest NREM sleep, the ones whose power on an EEG reflects the homeostatic intensity of the brain's recovery process, rise toward a peak across early childhood and reach their highest recorded values sometime in the first decade of life. The EEG of a sleeping 5-year-old shows delta waves of extraordinary amplitude. The peaks and troughs are steep. The slow oscillations sweep across the cortex with a force not seen at any other age, the electrical signature of a brain pressing its deepest restorative processes with the full urgency of growth. From this peak, slow wave power begins a long decline. The descent is slow enough to be invisible in any single year, but cumulative enough that across the span of decades, its magnitude becomes enormous. Between young adulthood and the 7th decade of life, the delta power measurable during NREM sleep is reduced by something approaching 3/4. The deepest valleys of the night grow shallower. The slow tide becomes a gentle ripple. Before any of this can happen, though, the sleep system needs a clock. The master clock of the human body is housed in a small paired structure just above the point where the optic nerves from both eyes cross on their way into the brain. It is called the suprachiasmatic nucleus. The name describes its location, above chiasm, nucleus, but the name gives no sense of the scale. Each half of the structure contains roughly 10,000 neurons. The whole thing, both halves together, is smaller than a grain of rice. And yet these neurons contain within themselves a molecular timekeeping mechanism that runs continuously, independently of any external signal, cycling through a complete oscillation in approximately 24 hours, day after day, year after year, for as long as the animal is alive. The mechanism inside each suprachiasmatic neuron is a loop of genes and proteins. Certain genes activate the production of proteins that, once they accumulate to a sufficient concentration, return to suppress the very genes that made them. The suppression holds until the proteins degrade, and then the genes activate again, and the cycle repeats. Each neuron in the nucleus runs this loop on its own, but the neurons are coupled to each other through signaling molecules that synchronize their individual rhythms into a single coherent population-level oscillation. The whole structure, in total darkness, with no information from the outside world, will cycle through its molecular clock with a period close to 24 hours, not exact, which is why entrainment by light is necessary, but close enough that the error accumulates slowly and the clock rarely drifts more than an hour per day from astronomical time. In a newborn, this clock is anatomically present. The neurons are there, the molecular loop is there, but the output is irregular, uncoupled from the day-night cycle in any reliable way. Melatonin, the hormone the suprachiasmatic nucleus drives the pineal gland to produce, a chemical signal that tells the body and brain that biological night has arrived, is not secreted in a clear nocturnal rhythm in the first weeks of life. The clock has not yet been entrained to the cycle of light and darkness it will use for every year that follows. The result is a sleeping infant who is not yet bound to the day, who drifts between sleep and wakefulness without clear reference to the sun, whose polyphasic bursts of sleep and waking follow an internal rhythm that has not yet synchronized with the outside world. Across the first 3 to 4 months of postnatal life, this changes. Melatonin secretion begins to consolidate into a clear nocturnal rise. The suprachiasmatic clock, receiving light information through the retinohypothalamic tract, begins to lock its oscillation to the cycle of day and dark that surrounds it. The baby's sleep, which had been scattered across the full 24 hours in roughly equal proportions, begins to gather itself toward the night. Not suddenly, gradually, across weeks, the nocturnal sleep periods lengthen, the daytime periods shorten, and the circadian rhythm that will shape every night's sleep for the rest of the life begins to exercise its organizing influence on the system. The suprachiasmatic nucleus and the ultradian sleep cycle are two different rhythms operating on two different timescales, but both are necessary for the sleep architecture that emerges by young adulthood to function properly. The circadian system sets the window, the broad span of hours during which the biological night is declared. Melatonin rises, core body temperature falls, and the conditions for sleep are created. The ultradian system fills that window, cycles of NREM and REM sleep cycling through their successive oscillations across the hours the circadian system has prepared. If either rhythm is disrupted, the other suffers. The clock and the cycle are not independent. They are interlocked, mutual, each depending on the other for the regularity and depth that healthy sleep requires. At the center of the NREM portion of each ultradian cycle sits the slow oscillation, the movement that defines slow-wave sleep most precisely. It is not simply a slow wave in the sense of a single electrical event at a low frequency. It is an organized alternation generated by the cortex itself between a state in which neurons across large areas of the cortex are simultaneously active, firing together, the electrical potential rising, and a state in which those same neurons fall briefly silent. The active state is called the up state. The silence is the down state. Together they produce the large, slow wave visible on the EEG. The crest of the wave corresponds to the up state, the trough to the down state, and the period of the full oscillation, crest to trough and back, runs at approximately half a cycle per second. This is a rhythm of extraordinary reach. The slow oscillation organizes other sleep rhythms around itself. Sleep spindles tend to appear during the ascending phase of the slow wave, as the cortex rises from its silent trough back toward the active crest. The thalamocortical circuits seize the moment to generate their brief, fast bursts of synchronized activity. The spindles are nested inside the slow wave, like smaller structures carried on a larger swell, and in the hippocampus, another fast oscillation, a brief ripple of neural activity associated with the replay of recent memories, tends to appear at the same moment during the ascending phase, when the cortex is briefly open and receptive. The slow wave, the spindle, and the hippocampal ripple arrive together in a coordinated sequence during the NREM sleep of a healthy young adult brain. This coordination is not incidental. It is the mechanism by which information moves across the sleeping hours from the short-term storage of the hippocampus to the longer-term distributed storage of the neocortex. All of this, the slow oscillation, the nested spindle, the coordinated ripple, begins in its most powerful form during childhood and declines from there. The 5-year-old's EEG shows slow waves of the greatest amplitude ever recorded in a living human brain. The trough of the down state is deep and long. The crest of the up state rises steeply. The spindles that nest within it are dense and frequent and well formed. The whole system operates at the height of its capacity, pressing deep into the restorative territory of slow wave sleep with an intensity that the adult brain will never match, and the aging brain will barely approximate. The arc begins there, at that peak, and the rest of the lifespan is the story of where it goes. The story does not move in one direction at a uniform pace. It has distinct chapters, each with its own character. The infant brain, still building its sleep architecture from the raw material of undifferentiated active and quiet sleep, is constructing the system from the inside out, the spindles appearing first, the slow waves consolidating, the circadian clock locking into its solar rhythm, the ultradian cycle stretching toward its adult length. Then the child's brain arrives at the peak of slow wave power during those years of early childhood and middle childhood when the delta waves are highest and the night's deepest sleep is its deepest possible. Then puberty shifts the timing. The circadian clock drifts later. The slow wave amplitude begins its long decline, the brain reorganizing its synaptic architecture under the pressure of developmental pruning. Young adulthood brings a sleep system that, relative to what comes before and after, represents something like a stable equilibrium, not the peak, but functional, deep enough, well organized, the spindle oscillation coupling still reasonably tight, the circadian rhythm still robust, the slow wave sleep still present as a meaningful fraction of the night. From young adulthood, the changes accumulate slowly. The slow wave percentage dips year by year, degree by degree. Sleep efficiency, the fraction of time spent in bed that is actually spent asleep, begins a gradual erosion. The spindles thin. The coupling between slow waves and spindles loosens. The circadian rhythm, that steady output of the suprachiasmatic nucleus, begins to show signs of flattening, its amplitude declining slowly over the decades, its entrainment becoming less precise. And in old age, all of these changes arrive at an endpoint that bears a strange resemblance to the beginning, a sleep that is shallow, fragmented, distributed across more variable hours, the slow waves nearly gone, the spindles sparse, the REM periods shortened, the architecture disrupted from both above and below. The return is not exact. The old person's sleep is not the infant's sleep. The infant was building upward toward the peak. The older person has come back down from it. But both, the newborn's rapid, immature cycling and the elderly person's shallow, fragmented night, represent states in which the fully developed, fully operational sleep architecture of healthy young adulthood is not yet or is no longer present. The system is always either on its way to something or coming back from it. The plateau, the peak, the years of fullest function, these are real, but they are not the default. They are an achievement of biological development, and their maintenance requires the right conditions, and their gradual loss is written into the aging process, as surely as anything else that changes across a lifetime. All of this plays out on the stage of the EEG. The traces are the record. A sleeping newborn's EEG looks different from a sleeping 5-year-old's, which looks different from a teenager's, which looks different from a 40-year-old's, which looks different from the trace of a person of 75. Each is recognizable as sleep, and yet each tells a distinct story about where in the arc the brain currently sits, the amplitude of the slow wave, the density of the spindles, the ratio of REM to NREM, the length of the ultradian cycle, the regularity of the circadian timing. These are not just technical parameters, they are a kind of record, a signature of the sleeping brain at a particular moment in its long existence. The suprachiasmatic nucleus sits above the optic chiasm in the dark of the skull, cycling through its molecular oscillation whether or not it is ever observed. Its roughly 20,000 neurons, 10,000 on each side, fire in coordinated waves, their individual clocks synchronized into a single population rhythm, the output of that rhythm driving the nightly rise and fall of melatonin, the nightly drop and rebound of body temperature, the nightly opening and closing of the window during which sleep is biologically possible. In a young adult, This signal is clear and strong and precisely timed. In a very old adult, it has grown quieter. The melatonin peak is lower. The circadian temperature rhythm is flatter. The clock is still running, but it runs with less amplitude, less certainty, a signal that has faded across the decades, like a voice speaking from a greater distance. Between these two ends of the arc, the same 20,000 neurons trace their molecular rhythm, unchanged in their basic structure, cycling through the same gene-protein loop that they have always cycled through, unaware of the decades passing outside the skull. The clock does not know how old it is. The rhythm does not know it is weakening. Each cycle simply follows the last, as it always has, at roughly 24 hours per oscillation, as long as there is life to sustain it. Sleep spindles, small and fleeting as they are, carry something of the same quality. On the EEG of a 30-year-old in deep sleep, they appear and disappear with a quiet regularity, waxing briefly, tapering, vanishing, replaced a moment later by the next. Each one lasts less than 2 seconds. Each one is the product of a thalamocortical loop, firing in its characteristic rhythm, 12 to 15 cycles per second, a burst of synchronized activity in the space between two slow waves. They are not dramatic. They do not announce themselves. They appear and go, again and again, night after night, doing whatever it is they do in the service of the sleeping brain. And the evidence suggests that what they do is important, that their presence or absence matters to the quality of the recovery the night provides, that the decline in their density across the adult years takes something away from the function of sleep that the lost hours alone cannot account for. The slow wave, though, is the fundamental rhythm. It is the thing from which the others take their organization. Half a cycle per second, the cortex rising and falling, the upstate and the downstate, the brief synchronized silence and the renewed collective firing. This oscillation is not unique to humans. Variations of it appear in the sleep of other mammals, in birds, in forms that diverged from the human lineage so long ago that whatever the slow wave does must be something very old, something that sleep discovered early in the history of nervous systems and has preserved ever since. Its peak amplitude in a sleeping child is the highest expression of something ancient, carried in the brain's architecture across an evolutionary timescale that dwarfs the individual lifespan entirely. And it is there, in this thought, that the full arc of a human sleep life becomes something worth resting with. Each night, across whatever age the sleeper is, the first months, the childhood years, the middle decades, the slower evenings of later life, the slow wave moves across the cortex at its unhurried frequency. In a newborn, it is just beginning to form. The precursor traces appearing in quiet sleep, the full oscillation still taking shape. In a child, it reaches its greatest amplitude, a tide at its highest mark. In a young adult, it persists, still deep enough to do its work, still well coupled to the spindles nested within it. And in the older decades, it retreats, lower, shorter, less frequent, until in some sleepers it is barely detectable, a faint motion in the electrical record, where once there was a deep sustained wave. The retreat is gradual. No single night is the last night of deep sleep. No particular year can be named as the point at which the tide turned. The change accumulates in the EEG records across populations, in the spectral power analyses of sleep researchers tracking the same individuals over years, in the quiet and mostly invisible arithmetic of delta power declining by fractions across each decade. From the outside, the sleeper looks the same, lying in the dark, breathing slowly, the body at rest. From inside the EEG record, the electrical character of the night tells a different story. one that has been telling itself without interruption since the first postnatal day and will continue telling it until the end. The slow oscillation itself does not know any of this. It moves because the cortical circuits that generate it are in the state that generates it, the neurons coupled, the homeostatic pressure sufficient, the conditions of the night fulfilled. Each sweep across the sleeping cortex is just the next sweep following the previous, as it has always done, with no knowledge of the sweeps that came before or the fewer that remain. A half cycle per second, the cortex rising, the cortex falling, the tide of electrical activity moving in the deep night across a sleeping brain, that this single oscillation will have a different character at 10 years old than at 30, and a different character again at 70, that the same electrical rhythm will trace its arc across a whole human life, rising to its peak and then declining through the decades in a slow curve that no single night makes visible, is one of the quieter, more enduring things that sleep science has revealed. The rhythm itself is ancient, the life through which it moves is brief, and somewhere in the space between those two scales, the deep evolutionary age of the slow wave and the single human lifetime across which it rises and falls, there is something that asks only to be noticed, and then very gently to be allowed to continue. A newborn lies still in the dim room, and then not still at all. The eyelids flutter, the lips pull and release in a slow, rhythmic purse, as though tasting something mild and pleasant in the air. One hand lifts slightly at the wrist and drops. The fingers curl and spread, and curl again, brief and automatic, while the rest of the body holds its long, soft weight against the mattress. The breathing shifts, faster, then uneven, then fast again, and the chest moves in a shallow, irregular tide that is nothing like the deep, slow breathing of a sleeping adult. This is not restlessness. This is not distress. This is a brain at work, doing some of the most consequential labor of its entire life, in a state that only resembles stillness from the outside. The twitches have a purpose that took a long time to understand. Each small, involuntary movement of the fingers, each brief flicker of the facial muscles, generates a signal, a proprioceptive echo, that travels upward through developing nerve pathways, arriving at the cortex like a message sent from the body's edges to its unfinished center. The cortex in a newborn is not yet a complete map. The regions that will eventually know where the hands are, what the face feels, how the limbs move through space, are still drawing their own borders. These small reflexive twitches, arriving in waves during what researchers call active sleep, help draw them. Each twitch is a calibration. Each involuntary curl of the fingers sends a faint signal upward into the unformed territory of the somatosensory cortex, where the message is received and the map, ever so slightly, becomes more precise. The neuroscientist Mark Blumberg has spent years documenting this process in developing rat pups, and what he has found appears to hold across species, including our own. In rat pups, as in human newborns, the limb twitches of active sleep are not random noise. They are patterned, they are generated by specific brainstem circuits, and they arrive at the cortex at exactly the period when sensory maps are most open to incoming signals. The twitching stops during wakefulness, when the motor cortex generates its own commands and the signals are no longer novel. It stops during quiet sleep, when the brain has shifted into a different register. It happens precisely and only during active sleep, in the window when the immature cortex is most ready to receive the information and most changed by its arrival. Active sleep and quiet sleep. These are the terms that apply in the first weeks and months of life, because the adult categories of NREM and REM sleep do not yet fully fit what a newborn's brain is doing. The electroencephalogram, the recording of electrical activity through the scalp, shows this clearly. In an adult, the NREM stages are marked by distinct spindle-shaped bursts of oscillation, by the tall, slow sweep of delta waves, by clear and reproducible signatures that a trained reader can identify at a glance. In a newborn, those signatures are absent or only just beginning to form. The EEG of a sleeping infant in quiet sleep shows instead a pattern called trace alternate, a series of high-amplitude bursts separated by intervals of relative electrical silence, like a signal arriving in packets rather than a continuous stream. It is the earliest precursor of the organized slow oscillations that will define deep adult sleep decades from now. But it is not those oscillations yet. The brain has not grown into them. Quiet sleep in the newborn carries its own stillness. The breathing slows and regularizes. The muscles hold their soft tone. The EEG, in its alternating bursts and suppressions, moves in a rhythm that has no precise adult equivalent, something between the silence of deep sleep and the beginnings of the organized waves that will eventually replace it. Then, within 50 or 60 minutes, the cycle turns and the brain slips back into active sleep, and the twitching resumes, and the eyelids flutter again, and the work of building the sensory cortex continues. A newborn does not move through sleep in the long, gradually deepening arc of an adult night. The whole cycle completes in less time than it takes an adult to reach the end of a first slow wave period, and then it begins again, and then again, in a rhythm that runs continuously through both day and night. That phrase, both day and night, is important. A newborn does not know the difference. The suprachiasmatic nucleus, the small cluster of cells above the optic chiasm that serves as the body's master clock, is anatomically present at birth, and its molecular gears are already turning. But the output of those gears, the rhythmic hormonal and neural signals that in a healthy adult coordinate the timing of sleep, wakefulness, temperature, and hunger across the 24-hour day, is not yet strong enough to organize behavior. The infant sleeps and wakes according to hunger, not according to light. The night is not yet different from the day in any way the young nervous system has learned to read. Sleep comes in short bouts, 4 or 5 or 6 of them spread across the full rotation of the earth, with wakefulness filling the gaps in no particular pattern that the clock or the sun has set. A 24-hour actogram of a newborn's sleep looks at first glance like a scatter of short dark marks against a pale field, blocks of sleep appearing at irregular intervals across both the daylit and dark halves of the day, with no clear preference for either. There is no long dark band of consolidated nocturnal sleep. There is no obvious rhythm. There is only the repeated, unanchored cycle of hunger and feeding and sleep and waking, distributed across all the hours without discrimination. The baby is not confused. The baby is simply operating on the system available, which is not yet the system it will eventually have. Between 14 and 17 hours of every 24 belong to sleep in these first weeks. The number is large by adult standards, and it needs to be, because the work being done in each sleep period is large by any standard. The twitching of active sleep builds cortical maps. The quiet sleep that follows it, with its alternating bursts and suppressions, consolidates whatever changes the active period made. The cycle turns. It happens again. The sleep of a newborn is not rest in the sense that adult sleep is rest, the clearing of the day's metabolic residue, the filing of memories, the slow release of accumulated fatigue. It is something prior to that. It is the construction of the system that will later, in a more grown brain, perform those functions. The amount of time spent in active sleep is striking. In a newborn, roughly half of all sleep time carries the signature of active sleep, the twitching and the irregular breathing and the flickering eyelids, all driven by circuits in the brainstem that generate the chaotic, rich internal activity the developing cortex requires. In a healthy young adult, the equivalent stage, REM sleep, occupies perhaps a fifth of the night. The difference is not incidental. The immature brain is not simply sleeping in adult patterns, with more REM added for emphasis. It is operating under a fundamentally different biological regime, one in which the demand for active, internally generated stimulation is at its maximum, because the structures that would otherwise provide stimulation through experience, through vision and touch and sound and movement in the waking world are not yet mature enough to do the job. The world slowly changes this. By the time an infant is 3 months old, something begins to shift. It is gradual, and it happens before it becomes visible in any behavior obvious enough to name. The melatonin rhythm, which in the first weeks is largely absent, begins to establish itself. The suprachiasmatic nucleus, receiving input from the eyes and responding to the pattern of light and dark, begins to coordinate its output more reliably. The hormonal signal of melatonin, low through the day, rising toward evening, peaking through the deepest hours of the night, begins to arrive at something approaching the adult pattern. The body starts learning, slowly, which half of the day is which, and the sleep begins to gather. The scattered distribution of short sleep bouts across all 24 hours starts to pull over weeks toward the nighttime half. The longest sleep bout, which in the earliest weeks might be 3 hours, might be 4, might arrive in the middle of the afternoon, begins to lengthen and to drift and to find the night. It is not a sharp transition. There is no single day when a 3-month-old decides to sleep through the night. The consolidation is a gradual biological process driven by the maturing of the circadian system and the slowly increasing capacity of the homeostatic sleep drive to accumulate and discharge across a longer uninterrupted stretch. But by around 3 to 4 months of age, most infants produce a longer nocturnal sleep bout for the first time, a stretch of 5 or 6 hours that falls in the dark half of the day, and that arrives with growing reliability as the weeks pass. The actogram of a 6-month-old tells a different story than the actogram of a newborn. The scatter is still there, especially in the daytime, blocks of sleep distributed across the morning and early afternoon, shorter now than they were in the earliest weeks, but still present. What is new is the long dark band of the night, against the remaining daytime nap blocks, which are still visible. The nocturnal sleep has lengthened and consolidated into something that looks in outline like the adult pattern. It is not yet the adult pattern. The total duration is still longer than an adult's typical night. Most 6-month-olds sleep 10 to 12 hours across 24, and the architecture inside those nighttime hours is still more active sleep-rich and cycle shorter than adult sleep. But the shape is there. The night has begun to mean something. Around 70 to 80% of infants sustain a 5 to 6 hour nocturnal sleep window by 12 months of age. The range is wide, and the variation is real. Some infants consolidate earlier, some later, and the factors that shape the timeline include not only the maturation of the circadian and homeostatic systems, but also feeding patterns, the family sleep environment, and individual differences in nervous system development that are not yet fully understood. But the direction is consistent. Across the first year of life, the biological pressure to consolidate sleep toward a single long nocturnal period grows steadily stronger, and the behavioral expression of that pressure, The gradually lengthening night, the gradually shrinking number of daytime sleep bouts, follows behind it. The naps themselves undergo their own quiet evolution. At 3 months, an infant might nap 3 or 4 times in the daylight hours, each nap brief, each preceded and followed by waking periods that are limited in duration by the young brain's limited capacity to sustain alert engagement. The total daily sleep of 10 to 16 hours is spread across all these periods, day and night, with the longest continuous stretch still modest by adult standards. By 6 months, the typical pattern has already condensed to 2 or 3 daytime naps, a morning nap, an early afternoon nap, and sometimes a brief late afternoon sleep. By 12 months, most infants have settled into a single midday or early afternoon nap, with the morning nap lost and the afternoon nap holding its ground. This one remaining nap is not simply a concession to fatigue. When a 12-month-old or 18-month-old sleeps in the early afternoon, the EEG shows something specific and rich, a concentration of slow-wave activity, high-amplitude delta oscillations moving through the NREM sleep of the nap at a density that reflects the homeostatic pressure that has accumulated across the morning's wakefulness. The slow waves of a toddler's afternoon nap are not pale imitations of nighttime sleep. They are dense with the same restorative delta activity that defines deep nocturnal sleep, compressed into a shorter window and discharged efficiently, so that the child wakes from the nap refreshed and ready for the afternoon's engagement. The nap is in a real biological sense a second sleep period, a supplementary discharge of homeostatic pressure that the young nervous system still requires, because its capacity to consolidate all of its slow-wave activity into a single nighttime stretch has not yet fully matured. The slow-wave system in a young child is operating at an intensity that will not be matched at any other point in the lifespan. The delta oscillations that define the deepest stage of NREM sleep, slow, rolling waves of electrical activity moving through the cortex at less than 4 cycles per second, generated by the coordinated rise and fall of millions of neurons shifting between their active and silent states together, are measurably larger in amplitude and more densely packed in a young child's sleeping EEG than in any other age group. The cortex is generating these waves with a force and regularity that reflects the strength of its homeostatic need. The young brain accumulates sleep pressure rapidly during its waking hours because it is building and connecting and reorganizing at a rate that no older brain can match, and the slow-wave sleep it generates in response is correspondingly intense. For a child of 3, 4, or 5 years old, the afternoon nap that remains in the daily routine carries a weight it does not seem to carry from the outside. Watching a 3-year-old sleep in the afternoon, curled on a mat, breathing slow and even, entirely still, there is nothing obviously remarkable about it. It looks like a child sleeping. Inside the sleeping brain, the first non-REM period of the nap produces a cascade of slow waves that move through the cortex in the long sweeping arcs characteristic of the deepest NREM sleep available to a human brain. The delta power in that brief sleep is in some measures among the highest that child's EEG will ever record. Not the highest in absolute amplitude. That title belongs to the overnight slow wave sleep of a slightly older child, at its peak in the years between 5 and 10, but dense enough and coordinated enough that the machinery of consolidation is fully engaged. The nap is also, in a practical biological sense, a buffer. The young brain cannot yet sustain all of its homeostatic recovery in a single nighttime stretch, not because the night is too short, but because the homeostatic pressure accumulated during waking accumulates too quickly and too intensely for a single long period of sleep to clear it efficiently. The nap is not a sign of weakness in the sleep system. It is a sign of the system's current operating range, the natural consequence of a cortex that is working so hard during the waking hours that it needs an intermediate discharge before the next full night of recovery can do its work. And then the nap goes. It does not go overnight, and it does not go all at once. Between 3 and 5 years of age, the afternoon nap begins to shorten, to shift later, and eventually to become intermittent, present on some days and absent on others, depending on the particular demands of the morning, the quality of the previous night, the temperature of the room, and then, on a day that is not marked by anything in particular, it does not come. The child plays through the afternoon. The child seems tired by early evening. The child sleeps that night with a long, dense first slow wave period that absorbs the homeostatic pressure the absent nap would have discharged, and the following morning the child wakes intact. The system has crossed a threshold. The single nighttime sleep can now carry the full weight. The disappearance of the nap is one of those transitions that attracts attention from families and caregivers, but almost no attention from the child. From the outside, it feels like the ending of something, a small milestone in a sequence of small milestones, the sort of change that gets noted and then forgotten because there is always a newer change arriving. From inside the biology, it is something more precise, the point at which the homeostatic slow wave system has matured enough to consolidate its full recovery into the nocturnal period, no longer requiring the supplementary discharge of the daytime nap. The cortex has grown strong enough. The night is now enough. This threshold is not reached uniformly. Some children retain a midday nap until 5, occasionally beyond, not because of any deficit in their development, but because the timeline is wide and individual variation is real. The homeostatic slow wave system is not a single switch that flips. It is a distributed network, maturing across years, and the point at which it becomes capable of full nocturnal consolidation is determined by the interaction of many factors: genetics, total sleep opportunity, the accumulation of developmental changes in prefrontal and hippocampal circuits that support the slow oscillations themselves. What is consistent is the direction. The naps grow shorter, there are fewer of them, and then there is none. By the time a child is 5 or 6 years old, the sleep pattern has a shape that is recognizably closer to the adult template than to the newborn's polyphasic scatter. A single consolidated nocturnal period, lasting 9 to 11 hours, carries the full burden of the night's recovery. The active sleep that dominated infancy has given way to a more balanced architecture, with NREM occupying a larger proportion of the night, and the slow wave stages within it, dense with the high-amplitude delta activity, characteristic of a young and vigorously homeostatic brain. The cycle length has extended. The sleep spindles, those brief, elegant bursts of thalamocortical oscillation that were largely absent at birth and began to emerge around 6 to 8 weeks of postnatal life, are now well established, generated reliably by the maturing circadian and thalamocortical circuits, and appearing consistently within the second stage of NREM sleep on every cycling night. The child sleeps in long, unbroken nights. The EEG of a sleeping 5-year-old, recorded across a full night, shows the pattern of NREM stages deepening in the first half of the night, and REM sleep gathering in the second half, the same alternating tide that will characterize adult sleep, but with the slow wave stages of the early night, richer and more sustained than anything that will be found at this depth again. The first slow wave episode of the night in a young child lasts longer and reaches a greater amplitude than the equivalent episode in a teenager, in a young adult, in a middle-aged person. The brain is generating slow waves with a force that reflects both the intensity of its developmental work during the waking hours and the vigor of a homeostatic system operating at close to its biological peak. In the quiet of a sleeping 6-year-old's room, the brain is completing what the twitching active sleep of infancy began. The cortical maps drawn by the proprioceptive signals of those early twitches are now refined by experience, by the accumulation of sensory encounters across thousands of waking hours, and the slow-wave sleep of each night continues the work of consolidating those accumulated changes, downscaling synaptic strengths that have grown too large, and stabilizing connections that the day's experience has selected. The slow waves are still building something. The process they serve has changed character from the raw constructive work of infancy, but the fundamental relationship between deep sleep and a developing cortex is continuous across all of it. The newborn twitching in active sleep and the 5-year-old lying still in the deep silence of its first slow wave episode are doing different work at different scales, but they are doing work biologically purposeful, developmentally consequential, measurably important to the brain that emerges on the other side of each night and each year. The sleep of early childhood is not passive. It is the most active thing the young organism does. There is something in the trace alternate of a quiet sleeping newborn, those alternating bursts and suppressions on the EEG, arriving in a rhythm unlike anything else in the sleep record, that is easier to understand once the full arc is visible. It is not an anomaly of immaturity. It is the earliest form of a system that will, over months and years, organize itself into the slow oscillations of adult deep sleep. The suppression intervals will narrow. The bursts will become more continuous. The slow waves will emerge from them the way a form emerges from what precedes it, gradually, without a single moment of completion, through the accumulation of small maturational changes in circuits that are growing steadily toward the patterns they will eventually maintain for decades. By the end of the first year of life, the trace alternate is gone. The slow-wave stages of NREM sleep have begun to take the form they will carry through childhood. By the end of the toddler years, the polyphasic scatter of the newborn's actogram has been replaced by the long dark band of a consolidated night. By the end of the preschool years, the nap has disappeared, and the single nocturnal sleep carries all the weight the system needs to carry. Each of these transitions is marked, from the inside, by changes in the EEG that are gradual and continuous, not steps but slopes, not thresholds crossed at a moment, but boundaries that shift over weeks and months, until one day the new pattern is simply what is there. The sleep of the very young is a process of becoming, not in the vague sense that all development is becoming. The word is used here to point at something specific. The brain that enters sleep at birth is incomplete in ways that sleep itself is completing. The architecture of sleep changes because the architecture of the brain changes, and the architecture of the brain changes partly because of what sleep is doing to it. And this circularity is not a logical puzzle, but a biological fact, as real as the twitching fingers and the trace alternate and the high delta power of the afternoon nap. Sleep and brain development are not parallel tracks. They are the same track, running in the same direction, at the same pace, for the same reasons. The afternoon nap of a 3-year-old ends. The small body stirs. The eyes open slowly, briefly unfocused, then clear. The waking comes gently, as it tends to in young children, not the sharp surfacing of an adult roused too early from sleep, but a gradual lightening, arising through shallow water toward the surface of the day. The afternoon stretches ahead. there are still hours of light. The homeostatic pressure that built across the morning has been partially discharged by the nap, and the system is steady, ready to continue accumulating through the afternoon until the evening deepens and the nocturnal period opens its long, slow wave rich first episode once again. A few years from now, the nap will be gone. The afternoons will run continuously from morning waking to the beginning of the night's sleep, and the first slow wave episode of the consolidated night will be the primary moment when all of the day's homeostatic accumulation is discharged. The brain will have grown strong enough to hold it all. The system will have matured to the point where the single long night is sufficient. But for now, in the still room, the 3-year-old wakes from the nap, and the afternoon continues, and the slow waves that moved through the sleeping cortex 20 minutes ago have done their work quietly without any witness. The brain built a little more of itself. The map shifted a little further toward the form it will eventually hold. The sleep was brief, and it was deep, and it was enough for now, for this age, for this particular and unrepeatable stage of the long, slow process of becoming a sleeping brain that will carry the night alone. On a school night sometime in late autumn, in a house where the lights have been off for an hour, a 10-year-old lies in the deep middle of sleep. The room is cool and dark. Outside, the wind moves through bare branches with a low, steady sound. Inside, the child's breathing is slow and even, the body almost perfectly still, and on a measure of electrical activity taken across the scalp, something extraordinary is happening. The waves are enormous. They sweep across the cortex in long, slow arcs, less than 4 times per second, their peaks rising high and their troughs falling deep, the kind of electrical sweep that represents the most powerful slow wave signal the human brain will ever produce across an entire lifetime. These are not the waves of a child who has simply fallen asleep. They are the signature of a brain at the absolute peak of its homeostatic power, generating delta oscillations with an amplitude and density that no later decade will match. The slow wave sleep of a 10-year-old is, by every measure available, the richest and the deepest sleep any human body will ever know. This is the baseline. Everything that follows, across the years that stretch from here through adolescence and into the long adult decades, is measured against this moment, the high water mark of a tide that from this point onward will only recede. The decline does not arrive all at once. It does not announce itself. It begins with the biology of puberty, which carries within it a set of changes to sleep so specific and so consistent that they have been documented across cultures, measured in non-human primates, and tracked through populations on several continents. What puberty does to sleep is not incidental, it is structural, and it begins with the very delta waves that were so abundant in the sleeping 10-year-old, sweeping, slow, and high, whose amplitude will start to fall before the child becomes fully aware that anything about the night has changed. The first thing puberty changes is the depth of sleep. In the years between early and late puberty, slow wave amplitude falls by something close to 40%. This is not a modest shift. On an EEG recording, it is visible as a gradual flattening of those high rolling oscillations, the peaks pulling back toward the baseline, the troughs rising to meet them, the whole electrical landscape of deep sleep becoming shallower by degrees. The researcher Erwin Feinberg, who spent decades studying the EEG of developing humans, interpreted this decline as an electrical record of something happening in the brain tissue itself. Across the years of puberty, the cortex prunes back the excess of synaptic connections that accumulated during childhood, a large-scale revision of the brain's wiring, cutting away the connections used least, and consolidating those used most. Slow wave sleep, in Feinberg's reading, is the EEG correlate of that pruning. As the synapses thin, the delta waves flatten. The two processes run in parallel, and they run for years. A cortical synapse map of the frontal lobe across these years would look like a field losing density. Early in adolescence, the connections are close and numerous, almost crowded against one another in the tissue. By the late teenage years, the same region is measurably sparser, not depleted, but refined, the way a tangled outline becomes a cleaner drawing. The brain that emerges from this process is different in kind, not just in degree. It is more efficient, more specialized, and as a measure of what it has done to get there, it generates less slow wave power across the night. This is not damage, it is development, but it means that the teenage brain is sleeping in a genuinely different way than the 10-year-old brain did, and the changes do not stop at the depth of sleep. Giulio Tononi and Chiara Cirelli, working from a framework they call the synaptic homeostasis hypothesis, have proposed that slow-wave sleep performs a particular task across every night. It downscales synaptic strength across the cortex, reducing the accumulated potentiation of the waking hours, resetting the system so that the next day can begin fresh. During adolescence, this process appears to intersect with the developmental pruning already underway. The nightly downscaling and the longer-term structural revision seem to share a substrate, running through the same tissue in the same window of time. Slow-wave sleep in this picture is not merely a passive reflection of synaptic change, it is one of the mechanisms through which the change is carried out. Each night of deep sleep in the teenage years is in some sense a session of architectural work, but there is a second change that puberty brings to sleep, and it is the one that most people notice. It does not show up on an EEG as a shift in amplitude. It shows up in the clock. The timing of sleep shifts later. This is not a preference. It is not a choice, and it is not a habit formed by staying up too late, looking at a screen. Those screens can make it worse. It is a biological event, driven by changes in the circadian system that are linked to the hormonal transitions of puberty itself. The dim light melatonin onset, the point each evening when the brain begins to secrete melatonin in response to falling light, signaling the biological start of night, shifts progressively later as puberty advances. In a 9-year-old, this onset arrives relatively early in the evening. In a 14-year-old moving through the middle stages of puberty, the same onset is measurably delayed, arriving later in the night by a margin that can be plotted on a graph and that grows across the pubertal years. The two curves, the child's and the adolescent's, diverge slowly and visibly on the same axis. Part of what drives this delay is a change in circadian period length. The internal clock of a pubescent adolescent runs slightly longer than it did in childhood, its natural cycle extending just enough that without external constraints, the whole biological day shifts later. Part of it is an increased sensitivity of the circadian pacemaker to light in the evening, meaning that exposure to light in the hours before sleep suppresses melatonin more strongly than it would have a few years earlier, pushing the biological night even further into what the clock on the wall calls late. And part of it is a change in the accumulation of homeostatic sleep pressure, the drive to sleep that builds across waking hours, which in adolescence appears to build more slowly than in younger children, so that the pull towards sleep arrives later in the evening and lasts longer into the morning. None of this is defiance. None of it is laziness or disorganization or a failure of will. It is a clock running on its own inherited time, shaped by forces that operate below the level of conscious choice. This phase delay has been documented in cultures that have no electric light. It has been measured in non-human primates who show the same pubertal timing shift as human adolescents. It is, in the language of biology, conserved, which means it appears across different species in different environments because it is written into the developmental program itself, not imposed by circumstance. What this means in practice is that the adolescent's biological night genuinely begins after midnight. The body does not want to sleep at 10 in the evening. The melatonin has not yet risen. The temperature has not yet fallen. The homeostatic drive has not yet accumulated to its peak. Asking a teenager in full puberty to fall asleep at 10 is something like asking an adult to fall asleep at 7 in the evening. The biology has not arrived there yet, and the hours of lying awake in the dark are not stubbornness but timing. The morning is the other side of the same problem. Because the biological night begins late, the biological morning arrives late. Waking at 6:30 or 7 in the morning, which is the normal schedule for most school systems, cuts the sleep period far short of its natural end. The adolescent who fell asleep at midnight and woke at 6:30 has had 6.5 hours of sleep. The adolescent whose natural wake time on a free morning might be 9 or 10 o'clock has had the last 2 to 4 hours of that natural night cut away. Those lost hours are not empty. They are disproportionately rich in REM sleep, the sleep stage that dominates the later cycles of the night, that is involved in the consolidation of emotional memory, in the processing of social information, in the regulation of the circuits that govern mood. Day after day, across the school week, those hours are removed, and then the weekend arrives, and the same body, no longer constrained by an alarm, sleeps until 9 or 10 or 11, following its clock back to where the biology wants it. The difference in sleep timing between a school night and a free night, measured in the same person across the same week, is the quantity that Till Roenberg and his colleagues have called social jet lag. It is not metaphorical. The same physiological disruption produced by flying across several time zones, the mismatch between the internal clock and the external schedule, is produced locally each week by the gap between when the adolescent's biology wants to sleep and when the school bell requires wakefulness. For some teenagers, this gap is small, an hour manageable, but for many, particularly those in the later stages of puberty, or those whose circadian clocks run naturally toward the later end of the population range, the gap is 2 hours, or 3, or more. A school night actogram and a free day actogram, placed side by side, show the same person sleeping at completely different times on different days. The school nights showing a compressed, late onset, early terminated sleep block, and the free mornings showing the clock snapping back to its natural position like a spring released. The offset between the two is measured in hours. The body experiences that offset as a weekly cycle of disruption and partial recovery. Ronneberg and colleagues followed this pattern across large populations and found that the consequences reach into every domain they measured. Higher levels of social jet lag, larger gaps between biological and school day sleep timing, were associated in their data with lower academic performance, with elevated rates of depressive symptoms, with higher body mass index, and with greater use of stimulant substances like caffeine. These are not small effects at the margins of significance. They form a consistent pattern across the data, and the pattern points in one direction: the chronic removal of sleep from its biologically correct window carries costs that accumulate across the school week and across the school years. This is worth sitting with in the slow way that allows it to become real rather than abstract. Somewhere in a school building in the middle of a weekday, there is a 16-year-old whose melatonin would not have risen until midnight, whose biological morning would not have arrived until 9, whose brain has been awake for 2 hours longer than its circadian timing would have allowed, and who is expected to learn, to concentrate, to consolidate what has been taught, to regulate their emotions through a long social day. The biology says this is difficult, not because the teenager is not trying, but because the clock governing every one of those functions is misaligned with the schedule by several hours. Growth hormone adds its own thread to this picture. Secreted in a large pulse during the first episode of slow wave sleep each night, growth hormone arrives in the early hours of the night in a sharp, high concentration, a secretory peak that is measurably larger during early puberty than at any other time in development. Plotted across a single adolescent night, the hormone curve rises steeply in the first hour of sleep, crests, and then falls back toward baseline, the whole event tied directly to the depth and intensity of that first slow wave sleep episode. The steeper the delta waves, the more robust the secretory pulse. This is one reason that sleep during the pubertal years matters not only for cognitive and emotional development, but for physical growth. The hormone that lengthens bones and builds tissue requires deep NREM sleep to reach the bloodstream in its full daily measure. The rising gonadal steroids of puberty, estrogen and testosterone, in their increasing concentrations, reach into the sleeping brain through several pathways. They modulate the serotonergic circuits that regulate mood and arousal. They influence GABAergic signaling, the inhibitory neurotransmission that helps the sleeping brain descend into and remain in deep sleep. They affect the generation of sleep spindles, those brief, symmetrical bursts of 12 to 15 cycles per second oscillation, nested within the flatter terrain of NREM sleep, whose density and timing begin to shift across the pubertal years in patterns that differ between males and females. The adolescent brain is not the same biochemical environment as the child's brain, and sleep within that environment reflects the difference. All of these changes happen below the level of what any teenager could observe or report in ordinary language. What they might notice is that they feel more awake late at night than they used to, that mornings are harder than they remember mornings being, that on weekends, given the freedom to follow the clock wherever it leads, sleep arrives late and extends far into the morning, not as excess, but as return, the body taking back what the school week removed. These experiences are accurate. They are not distortions or complaints. They are reports from a body navigating a genuine biological shift, as real and as involuntary as the height that is changing, the voice that is changing, the body that is reorganizing itself around a new hormonal landscape. The change in circadian timing does not peak and then reverse immediately at the end of adolescence. In most people, the phase delay reaches its maximum somewhere in the late teenage years or very early 20s. The exact timing varies across individuals, and there is substantial natural variation in the population, with some people carrying clocks that run early and some that run late throughout life. But the pubertal delay is a shift on top of whatever baseline the individual carries, and as the hormonal transitions of puberty complete themselves, the clock begins, slowly, to track back toward earlier timing. This reversal is gradual. It takes years. Unwandel, and throughout it the slow wave decline that began with the first stages of puberty continues, the delta amplitude falling across the late teens, the 20s, the early adult decades, following a trajectory that has been set in motion and will not reverse. The rich, high-amplitude slow wave sleep of the 10-year-old on that autumn night, recedes further with each passing year, not in any single dramatic collapse, but in the slow, continuous way that a tide pulls back from a shore it has already reached, leaving behind a different landscape, still navigable, still productive, but changed. What the adolescent brain is doing during all of this, the pruning, the phase delay, the hormonal modulation, the weekly cycles of social jet lag, is becoming the adult brain. The process is neither comfortable nor tidy. It runs for years. It requires sleep to do its work, and it disrupts the very sleep it needs. There is something both difficult and ordinary about that. The biology does not resolve the tension it creates, it simply runs its course. The prefrontal cortex, the last region of the brain to complete its developmental arc, the tissue that governs impulse regulation, long-range planning, and the modulation of emotional response, is still maturing through the early 20s, its synaptic density still adjusting, its connectivity with subcortical structures still refining. The slow-wave sleep that accompanies this protracted maturation continues to serve the same function it served in earlier childhood, supporting the overnight downscaling and consolidation that the Tononi-Sorelli framework describes. It does this at a lower amplitude than a decade earlier. It does it across a compressed and often disrupted night. It does it, in many cases, while the homeostatic clock and the school or work schedule pull against each other with a force measured in hours, and still the development proceeds. The brain completes itself. The clock settles, gradually toward a timing that will remain relatively stable for several adult decades before the arc turns again. The synaptic architecture of early adulthood, refined through the work of 10 years of slow-wave sleep and pubertal pruning, becomes the foundation on which the rest of a cognitive life is built. There is something slow and quiet in that. The architecture is not built in an afternoon. It is not assembled in a season. It is built across a decade of nights, each one contributing a small increment of consolidation and revision, the cumulative result visible only in retrospect, in the person who wakes one morning in the middle of their 20s and finds that the world feels, in ways they cannot quite specify, slightly more legible than it did before. The delta waves that made that possible are smaller now. The slow oscillations still move across the sleeping cortex, but the peaks are lower, the troughs shallower, and the total energy in the slow wave frequency band is a fraction of what it was when the child was 10 and the waves were vast. Something has been spent to make this. That is not a loss, exactly. It is the cost of becoming. On a free Saturday morning in late winter, in a house where the curtains are still drawn well past 9, a 17-year-old lies in the slow, extended pull of a sleep that has finally been allowed to run its natural course. The room is warm. The light outside is pale and cold, pressing gently at the edges of the curtains without breaking through. The breathing is deep and even. The clock on the wall says 9:45, which is, for this particular biology, only an hour or so past what would have been the natural midpoint of the night. The melatonin rose around midnight. The deep slow-wave sleep came in the first 2 cycles, doing its work of consolidation and revision in the compressed window available. And now, in the later cycles of this extended morning sleep, REM has taken over, longer and richer with each successive cycle, the brain replaying and reorganizing the emotional texture of the days just past, processing the social world through the particular chemistry of the dreaming state. These are not wasted hours. They are the hours that the school week removed, and the body is reclaiming them with the efficiency of a system that understands, below any level of thought, exactly what was missing and what form of return is needed. The biological night in this body is not over. The clock that governs it reads differently from any clock on a wall, and the sleep that is happening now at 9:45 on a Saturday morning with the pale winter light held back by curtains, this is not laziness or waste or adolescent excess. It is a clock running on its own inherited time, following a rhythm that has been built across the years of development, shaped by hormones and by genes and by the long arc of a biology that is still, even now, in the slow process of becoming what it will be. The melatonin will fall toward morning, the cortex will brighten, the 17-year-old will wake in an hour, or perhaps two, and the day will begin. And on Sunday night, the alarm will be set again for 6:30, and the biological night will not have arrived by the time the house goes dark. And the clock will do what it does regardless, beginning its slow melatonin rise well after midnight, carrying the sleep period into the hours that the school morning will cut away. The tension is real. It is not invented, and it is not cured by willpower. It runs on biological time, which answers to its own calendar, and it has been running in adolescence since long before there were schools, or alarm clocks, or any schedule at all that the external world imposed on the internal one. What the delta waves record, in the hours that sleep is allowed to continue, is still the same fundamental thing they recorded in the 10-year-old in autumn: a brain at work, oscillations moving across the cortex in slow, organized sweeps, synaptic connections being evaluated and revised, memories being consolidated from one form of storage into another, the homeostatic pressure of the day being bled away hour by hour until the system is ready again. The amplitude is lower than it was. The timing is later. The architecture of the night has changed shape in ways that will continue to change for decades to come. But the slow wave is still there, still moving, still doing the only work it has ever known how to do. It moves through the sleeping brain the way a long tide moves through a quiet bay. Not fast, not dramatic, barely perceptible from the surface, sweeping through the synaptic landscape in the dark, leaving things slightly different than it found them, and then pulling back and coming again. The wave crests, the trough follows, and in the pale, still space between one oscillation and the next, somewhere in the pause at the bottom of the cycle, the adolescent brain continues its slow, invisible work, pruning and consolidating, revising and settling, in the only window of time the biology has always known was made for this. The curtains hold the light. The breathing continues, slow and even. Outside, the winter morning is cold and quiet, and the world waits without impatience for the sleep to be done. A 25-year-old lies down in the first hour after midnight. The room is dark, the body is still, and somewhere in the first 90 minutes of sleep, something extraordinary happens that most people will never know is happening at all. The slow waves come first. They build from the prefrontal cortex and roll backward across the sleeping brain, in long, unhurried sweeps, each oscillation lasting roughly 2 seconds, each crest and trough a coordinated silence, and then a sudden surge of electrical activity moving through tens of millions of neurons at once. On the scrolling trace of an EEG, they look like mountains drawn by a slow hand, broad, symmetrical, their amplitude higher than anything else the waking brain produces. This is slow-wave sleep at its young adult peak, dense, efficient, and generating the deepest physiological rest the body will reach all night. The heart rate has dropped, the muscles have released, the temperature at the core has fallen by nearly 2 degrees Fahrenheit. And for this brief window, this first long episode of deep sleep, the architecture is working almost exactly as it did in childhood, when these same waves were even larger, even more sustained. The slow waves will not stay this large. The body does not know this yet, but the change is already underway. Deep inside the cortex, in the years between 20 and 30, the homeostatic machinery that generates slow wave sleep has already begun a gradual drift. Not a breakdown, not even a fault, not anything that registers as disease. Just a quiet biological drift, measurable on EEG, if anyone happens to be looking, but felt by almost no one. The slow oscillations that define the deepest sleep begin to lose a small amount of power with each passing year. The peaks are still there, the waves still come, but the energy within that slow wave frequency band, the range between half a cycle and 4 cycles per second, begins to fall, and the fall does not stop. By the time a person reaches 50, the slow wave sleep that made up roughly a fifth of the night at 25 has contracted to somewhere between 5 and 10% in men. The spectral EEG plot tells the story plainly. The delta peak, which in youth rose steeply from the baseline, now sits flatter, its highest point pulled downward as though the signal has been slowly drained. The waves are not absent, the sleep is still real, but the deepest tier of the night, the biological bedrock that the young brain descends into so reliably, has grown thinner, and the sleeper rests in lighter terrain. This is the slow drift of midlife sleep. It begins before most people think to look for it. It moves too gradually to feel like loss, and for a long time it leaves almost no trace in the ordinary experience of being awake. The drift does not happen equally in everyone. Women and men sleeping side by side in the same decade of life carry measurably different sleep architectures. A woman in her 40s generates more slow-wave sleep than an age-matched man, more delta power across the night, higher amplitude oscillations, a thicker and more sustained descent into deep NREM sleep. Spectral EEG plots from large research samples confirm this consistently. The woman's slow-wave curve sits higher across the frequency range. The gap between the two lines quiet but persistent, a record of a biological difference that has accumulated since the earliest adult years. And yet, in population surveys, women report sleeping worse. They describe their sleep as lighter, less restorative, harder to obtain. They are more likely to lie awake in the middle of the night and more likely to report the experience of sleep as something precarious, something that can be lost. The EEG says one thing, the experience says another. Both are true. What the EEG cannot easily capture are the other forces acting on sleep from outside its own architecture. Insomnia, which affects women at roughly twice the rate it affects men across the adult decades, does not necessarily destroy slow-wave sleep. It fragments it, wraps it in anxiety, and makes the night itself feel unsafe. Restless legs, more common in women and worsening through pregnancy and beyond, introduces an irritant into the descent towards sleep that no amount of delta power can fully overcome. Mood disturbances leave their own mark. The sleep that the EEG measures is not the same thing as the sleep that is felt, and for women in midlife, the gap between those two things can be wide. There is something worth sitting with in that gap. A brain generating abundant slow waves and still waking at 3 in the morning, still lying in the dark with the night spread out around it, still arriving at morning feeling hollowed. The architecture is there. The depth is there. Something else is wrong, and it is not the kind of wrong that a spectral plot can name. The slow-wave decline in men follows a steeper path. By their mid-40s, many men spend proportionally far less time in the deeper NREM stages than women of the same age. By 50, the descent into N3, the slow-wave stage, is sometimes brief enough to register only at the edges of the hypnogram, the body still cycles. The ultradian rhythm still carries the sleeping brain between lighter and deeper sleep across the night. But the deepest point of each cycle has moved upward, and the night, measured in the currency of true slow wave depth, has grown shallower without announcement. The body notices this even when the mind does not. Sleep efficiency, the ratio of actual sleep to time spent in bed, begins a gradual decline across the adult decades. At 25, a healthy adult sleeps for something close to 95% of the time they spend in bed. By the mid-50s, that proportion has drifted to somewhere around 80 or 85%. The difference does not sound like much. 15 minutes awake in every hour and a half of lying in bed. A few more minutes at the surface before the next descent. A slightly earlier final awakening that cannot quite be slept back through. But the arithmetic of 100 nights, of 1,000 nights, adds up to something that the brain registers even when the waking mind does not. A gradual uncoupling of rest from the time set aside for it. The night grows slightly less reliable. The bed, which at 25 absorbed the body and returned it to sleep without incident, requires a little more negotiation by 50. The change is not only in the depth of sleep, it is in the timing. And the timing across midlife begins to shift in a direction most people notice only as a quiet inconvenience, the tendency to wake earlier than intended, to find the last hour of sleep too light to recover, to feel a pull toward sleep in the evening that arrives before it is welcome. The circadian signal has not failed, but it has begun, very gradually, to move forward in time, and the night it governs is growing slightly shorter at its far end. Inside this gradual change in depth and timing, one mechanism in particular carries consequences that ripple outward far beyond how rested a person feels in the morning. It is a mechanism so precise that it requires two different neural rhythms to work in exact coordination. Two electrical events timed to within a fraction of a second of each other, night after night, for the transfer of what the day has left behind in one part of the brain to be made permanent in another. The slow oscillation comes first. It rises from the cortex, a long, smooth sweep of synchronized neural silence, followed by a burst of firing, the full cycle taking about 2 seconds. And nested within the rising phase of that oscillation, timed to land precisely on its upward slope, is a sleep spindle, a brief, dense burst of 12 to 15 cycles per second, waxing and then waning over less than 2 seconds, its spindle shape unmistakable on the EEG trace. These two rhythms, the slow oscillation and the spindle, do not simply coexist. They couple. The slow wave orchestrates the spindle's timing, and the spindle, locked into the right moment of the slow wave cycle, opens a narrow window in which memory traces held in the hippocampus can be transferred to the longer-term storage of the neocortex. This is the overnight mechanism by which the sleeping brain consolidates what the day has taught it, not by rehearsing it, not by replaying it in narrative form, but by coordinating these two electrical rhythms across thousands of coupled events per night, each one brief enough to be invisible, each one contributing a small packet of information to the gradual construction of permanent memory. In a young brain, this coupling is precise. The spindles land on the right phase of the slow wave, consistently, reliably, with the timing that the transfer requires. In the aging brain, even in the moderately aging brain of a person in their mid-40s or early 50s, this coupling loosens. The two rhythms continue. The slow waves come, shallower but still present. The spindles come, still spinning up their brief bursts of sigma band activity. but the temporal relationship between them drifts. The spindles land slightly off the ideal phase. The window for hippocampal to cortical transfer opens at the wrong moment, or incompletely, or less often, and the overnight consolidation of memory, the silent sleeping work that turns the experience of a Tuesday into something retrievable on a Wednesday, grows less efficient. This is not the forgetting of aging as it is commonly described. It is not the dramatic erosion of recall that comes with disease. It is something quieter, a slight thinning of the night's ability to do what it does, a small reduction in how fully the sleeping brain can close the loop between learning and remembering. The memories that do not consolidate well are not lost dramatically. They simply fade a little faster, stick a little less firmly, require more effort to retrieve than they should. It feels like the normal imperfection of memory. It is, in part, the imperfection of sleep, the slow, coordinated loss of spindle oscillation coupling across the adult decades means that two people, one at 25 and one at 55, can spend the same number of hours in bed, report roughly similar total sleep times, and arrive at the following morning with very different degrees of overnight consolidation. The 55-year-old has slept. The architecture is there. The cycle's completed. The morning arrived. But the internal mechanism, the tight coordination of two rhythms that together accomplish the night's most precise neural work, has drifted, and the drift has a cost that accumulates over years. Outside the bedroom, across the industrialized world, roughly 1 in 6 workers faces a different kind of disruption entirely. Their problem is not the gradual aging of a biological clock, but the wholesale misalignment between that clock and the demands of a work schedule that runs against the body's own time. Shift work, the broad category of schedules that require regular work during the body's biological night, is not a marginal condition. It is the daily reality of workers across healthcare, transport, manufacturing, emergency services, and a dozen other industries. For the person whose shift begins at 10 in the evening and ends at 6 in the morning, the body's circadian system is running on its preferred schedule, preparing for sleep when the person needs to be alert, withdrawing melatonin and raising core body temperature just as the shift ends and the person attempts to sleep. The clock and the schedule are pulling in opposite directions, and the clock cannot simply be adjusted. A week of actigraphy from a rotating shift worker tells this story without words. 7 rows of data, one for each day, showing when the body was active and when it rested. In a person sleeping on a normal nocturnal schedule, those rows would show a consistent dark band of sleep, occupying roughly the same portion of each 24-hour line. In the shift worker's chart, no such band exists. Sleep appears at different times on different days. The block that represents Monday's sleep sits in the middle of the afternoon. Wednesday's is carved from the morning. The free days show the body attempting to return to biological night, but the return is incomplete, the sleep still fragmented, still landing in terrain that the circadian system has not fully prepared. The internal clock and the behavioral schedule never settle into agreement. The body that sleeps against its own clock does not simply sleep at the wrong time, it sleeps differently. Slow-wave sleep, which depends in part on the homeostatic pressure that accumulates across waking hours, arrives at a lower depth when that waking period was spent in biological night rather than biological day. REM sleep is curtailed when sleep is forced to begin before the circadian system has completed the transition toward biological morning. The sleep is real, but it is architecturally compromised, a building constructed with the correct materials in the wrong order, missing structural elements that would have appeared if the timing had been right. Over months, then years, then decades, the chronic misalignment between circadian time and behavioral time compounds in the body in ways that extend well beyond tiredness. The physiological systems that run on circadian rhythms, glucose regulation, cortisol secretion, immune function, cell division, are coordinated with each other by the shared timing signal of the circadian pacemaker. When that signal is perpetually misaligned with the actual pattern of light, food, and activity, the coordination between these systems degrades. The metabolic machinery begins to run less cleanly. Insulin sensitivity drifts. Cardiovascular regulation loses some of its smooth daily rhythm. And across large populations, long-term shift work is associated with elevated rates of metabolic syndrome, cardiovascular disease, and certain cancers, not because shift work is exotic or extreme, but because the body's systems were built to work in time with each other. And they work less well when they can't. These are slow consequences. They do not arrive with a clear announcement. A shift worker in their 30s feels tired, adjusts, manages, the metabolic drift is invisible, the cardiovascular pressure barely registers. And then a decade passes, and the cumulative weight of the misalignment shows up in a blood panel, or a clinic visit, or in the shape of a body that has aged slightly faster than its chronological years would predict. The cost is real. It was always real. It simply took long enough to accumulate that there was no single moment at which it became visible. The midlife bedroom, then, holds many kinds of sleepers. Some are drifting through the gradual architectural thinning that comes with age alone. Some are absorbing the additional burden of a circadian schedule that the rest of the world has organized against them. Some are lying awake with a brain that generates adequate slow waves but cannot rest in them. And some are sleeping deeply enough by every physiological measure and still arriving at morning, feeling that the night did not do what it was supposed to do. The mechanism that connects these experiences, the common thread running beneath all of them, is the slow oscillation. That long measured sweep of coordinated neural activity, rolling across the cortex at half a cycle per second, is the substrate on which so much of the night's work depends. Memory consolidation rests on it. Glymphatic clearance runs fastest during it. Growth hormone peaks within the first episode of it. The body's deepest physical restoration correlates with its presence and depth. And it is this rhythm, this specific, measurable EEG visible rhythm, that midlife presses against most consistently, from the inside through the gradual loss of homeostatic drive, and from the outside through the scheduling pressures that prevent the body from reaching the conditions it needs to generate it. A spectral EEG comparison between a 25-year-old and a 50-year-old, displayed side by side, carries a kind of quiet eloquence. Both traces are real sleep. Both contain delta activity. Both show cycling, recovery, the architecture of a functioning sleeping brain. But in the lower frequency range, the band below 4 cycles per second, where the slow oscillations live, the 50-year-old's curve is flatter. Its peak is lower. The energy that at 25 filled the slow wave band with a dense concentration of power has spread and thinned across the years, redistributed somehow across lighter frequencies, or simply reduced. The trace is not broken. It is older. The same instrument, played with less amplitude, producing a quieter version of the same fundamental note. What is striking is how long this change takes to produce any experience that could be named. The spectral change is measurable in the mid-30s. The consolidation effects are documentable in the 40s. The sleep efficiency decline is statistically significant by the mid-50s. And yet, most people living through these decades describe their sleep as mostly adequate, occasionally troubled, and only rarely as something they have thought carefully about. The change is too slow for any single night to reveal it. It has to be measured across years, across populations, by someone who is specifically looking for it. To the person living through it, the gradual loss of deep sleep across midlife is nearly invisible, a drift so incremental that each individual night feels more or less like the last. This is not a reassurance. The invisibility of the change does not mean the change is small. It means it is slow, and slowness, across the arithmetic of midlife, still arrives somewhere. There is a particular quality to the knowledge that something important is changing in the sleeping brain across the adult decades, not rapidly, not catastrophically, but persistently and measurably, and that the change is connected to things that matter far beyond the morning's mood. Memory, metabolic health, the long arc of brain aging. These are not abstractions, they are the lived experience of the years ahead, being quietly shaped by the quality of the nights already past. The slow oscillation moves across the sleeping cortex at roughly one sweep every 2 seconds. Its peak voltage, recorded on an electrode at the scalp, is the highest electrical signal the brain produces in any state other than certain forms of seizure. At 5 years old, those peaks are enormous, sweeping, high amplitude, the markers of a homeostatic system working at full strength to consolidate the staggering amount of learning and growth the young brain undertakes each day. At 25, still large enough that the trace looks like a range of low hills, measured and steady. At 50, lower, the hills are still there, but worn, their contours softened, the highest points no longer quite so high. None of this is disease. The decline in slow wave power across the adult decades falls within the range of typical biological aging. It is not a disorder, not a failure, not an emergency. It is what happens to a brain that has been awake for decades, that has been carrying its architecture through the gradual changes that time brings to all biological systems. The slow oscillation is not failing. It is aging, the same way the body ages, not in a single moment, but in the long continuous drift of biological time. And yet within that drift, within the ordinary aging of a mechanism that almost no one knows they have, something important is gradually changing in what the night can do. The tight coupling of spindles to slow waves, which at 25 operates with a precision that feels engineered, the two rhythms locking together like gears, by 50 operates with slightly more play in the mechanism, not broken, not even obviously impaired, just slightly less precisely timed, and the precision is what the function requires. A 50-year-old brain that could regain that coupling, that could return even a fraction of the tight spindle oscillation timing of a younger brain, would consolidate more from the previous day, clear more metabolic waste in the night, arrive at the morning with more of what the night was built to provide. The biology is not entirely fixed. The mechanisms are still present. The architecture is still cycling. What has changed is the precision of the coordination between its parts. And precision in biological systems is not always as lost as it appears. For now, in the midlife bedroom, the body rests, the cycles turn, the slow waves come and go, quieter than they were, but still present. The spindles still nest within them, imperfectly coupled, but still firing, still generating their brief bursts of sigma band rhythm. The hippocampus still sends its memory traces upward toward the cortex. Some of them arrive, most of them arrive. The night still does most of what it has always done. The room is dark and warm, the breathing has slowed, the heart beats steadily in its old reliable rhythm, slightly slower than it was an hour ago, slightly slower than it was at midnight. The sleep continues, deep enough to matter, lighter than it once was, carrying its quiet architecture into the hours before dawn. Somewhere in the low frequency range of the sleeping brain's electrical life, a slow oscillation crests and then releases, a long, smooth wave rolling across the cortex in the dark, and nested on its rising slope, just briefly, the tight, bright cluster of a spindle. The two rhythms, for that moment, are synchronized. The window opens, the transfer happens. Something learned yesterday is for this small fraction of a second being made permanent. The spindle fades, the slow wave completes its trough and begins to rise again. The night continues around this small precision, this brief coupling of two rhythms in a sleeping brain, two oscillations that together have been doing this same work in this same way since the first years of life. A little slower now, a little less tightly wound, but still turning, still meeting, still doing in the quiet of the night what the waking hours cannot do alone. The slow wave moves across the cortex and settles. The spindle comes and goes. The night holds everything in its dark, steady arms, and the brain, in its careful and imperfect way, continues the long work of remembering what it means to have lived another day. In a small paired structure no bigger than a grain of rice, nestled just above the place where the optic nerves cross inside the skull, something is changing. It has been changing for decades. The neurons there still fire in their ancient daily rhythm, still carry the signals that organize the body's sense of day and night, but they fire more slowly now, more faintly, their coordinated pulse grown softer with each passing year. The melatonin signal that rises from this rhythm used to climb steeply in the evening hours, a clear hormonal tide that swept through the blood and told every organ that the night had come. In older age, that tide still rises. It simply does not rise as high or as fast or hold its crest as long as once it did. The master clock has not stopped. It has grown quieter, the way a bell grows quieter when the metal thins, the shape of the sound still there, still recognizable, but carrying less far into the dark. This dimming of the circadian signal is where the story of sleep in older adulthood begins. Not with a sudden change, not with a clear boundary the body crosses, but with a gradual softening that has been accumulating across the decades, each year adding its small increment to a process that began, barely perceptibly, long before old age arrived. The suprachiasmatic nucleus, that paired cluster of roughly 20,000 neurons that has been generating daily rhythms since before birth, loses neurons across the lifespan. The vasopressin expressing cells within it, a population whose coordinated firing helps amplify and coordinate the daily signal, are fewer in number by the 7th decade than they were in the 4th. The nucleus's responsiveness to light, the primary signal by which the clock resets itself each morning to the turning world, grows blunted with age. Dimmer light enters through aging lenses and less responsive retinal cells. The signal that reaches the clock is weaker. The clock, in turn, sends a weaker signal outward, and so the architecture of the day, the rising and falling of alertness, the timing of hunger and temperature and cortisol, and the deep nocturnal slope toward sleep grows shallower, less defined, less reliably anchored to any fixed hour. One consequence of this flattening is a shift in the timing of sleep itself. When the circadian signal weakens, the evening rise of melatonin tends to move earlier. The window of biological night advances toward the afternoon and retreats before dawn. Many older adults find themselves growing sleepy in the early evening, then waking in the early morning hours, when the body's internal clock has already begun its daily rise, long before the first light appears at the window, and long before the world considers it a reasonable hour to be awake. This is not poor sleep discipline. It is not weakness or anxiety or failure of the will. It is the clock running on its own changed time, its evening bell ringing earlier than it once did, its morning signal arriving before the sky has begun to pale. The rhythm that once swept in a tall, clean arc across the night has grown broader, lower, less precisely bounded, and the sleep it organizes reflects every one of those changes. If the melatonin curve tells one part of the story, the overnight record of brain electrical activity tells another. In an older adult sleep laboratory recording, the trace of slow-wave sleep has changed profoundly from what it was in young adulthood, and more profoundly still from what it was in childhood. The large sweeping delta oscillations that once rolled through the first deep NREM episode of the night, the same waves that carried their greatest amplitude in childhood, still considerable in young adulthood, are smaller now. In many men and women past the age of 70, the stage of sleep defined by these slow waves barely appears at all. Polysomnographic records from healthy older adults routinely show that the deepest stage of NREM sleep, the stage once called slow-wave sleep, occupies less than 5% of the night's total sleep time, and in some individuals, nothing measurable remains. The waves are not merely smaller. The homeostatic process that generates them, the pressure that builds across each waking day and discharges overnight in a tide of slow oscillations, has itself been altered. The overnight decline of delta power that marks healthy slow-wave sleep regulation, the steep fall that should follow a night's deep sleep and flatten by morning, is blunted in older age. The regulatory mechanism is not simply producing less slow-wave sleep. It is producing it less efficiently, with less precision, as though the system that once calibrated itself each night against the demands of the waking day can no longer read those demands as clearly. What remains instead is sleep that is lighter, more interrupted, more easily displaced. The hypnogram, the overnight map of sleep stages, looks different in an older adult, not only in the depth of each stage, but in the stability of each transition. Where a younger person tends to cycle through NREM and REM sleep in relatively long, unbroken arcs, each stage holding its depth for an extended period before the cycle lifts toward waking and descends again. The older adult's overnight map shows more transitions, more frequent brief arousals, more time in the lighter stages of NREM, where the boundary between sleep and waking is thin and easily crossed. Sleep becomes in this sense more porous, not absent, not even necessarily shorter in total duration, but more permeable, the boundary between sleeping and waking less firmly held, the night more easily interrupted by noise, by temperature, by pain, by the simple fact that the mechanisms that enforce and deepen sleep have changed their character across the years. Some of this change is purely architectural. The thalamocortical circuits that generate sleep spindles, those brief rhythmic bursts of electrical activity that help gate the brain against sensory intrusion during sleep, produce fewer spindles in older age, and the spindles they do produce are smaller in amplitude, shorter in duration, and less precisely timed. The slow oscillation that in a younger adult rises across the sleeping cortex and carries within its ascending phase a precisely timed sleep spindle, the two rhythms interlocked in a brief coordinated event, is less tightly coupled in the older brain. The coordination that makes NREM sleep an efficient machine for certain kinds of consolidation and restoration becomes with the years progressively less precise. The parts still work. The timing between them has shifted, And the consequences of that shift across decades are not small. Somewhere in the fluid that fills the spaces around the brain's neurons, a clearance process is running. It runs in the dark, during sleep, in the deep quiet of slow-wave NREM. It involves the movement of cerebrospinal fluid along channels that run beside the blood vessels, threading through brain tissue, fine pathways lined with the end feet of astrocytes, those star-shaped support cells whose aquaporin 4 channels act as gates for fluid movement. During waking, these channels carry some fluid. During sleep, specifically, during the slow oscillations of deep NREM, the space between neurons expands by roughly 60%, and the rate of cerebrospinal fluid movement through the perivascular channels increases dramatically. The result is something like a slow tide moving through the brain's tissue while the neurons are quiet, not a fast flush, but a steady, gentle clearing of the molecules that have accumulated in the interstitial space across the waking hours. Among the molecules cleared by this process are two proteins whose names have become familiar in the context of one of the most feared diseases of older age. One is amyloid beta, a small peptide that in high concentrations aggregates into the plaques found in Alzheimer's disease. The other is tau, a protein that in altered form forms the neurofibrillary tangles that define the same disease's progression. The glymphatic system, the name given to this astrocyte-mediated clearance pathway, moves both outward through the brain's tissue, while slow-wave sleep holds the cortex in its long rolling oscillations. This is what the deep sleep of any night has always been doing, in every brain, across every age of a life. It is a maintenance function, ancient and essential, operating far below any threshold of experience or awareness. The relationship between this clearance system and the aging of sleep is not a simple one-way line. Slow-wave sleep diminishes with age, and with it the glymphatic clearance that depends on its oscillations. Amyloid beta and tau proteins accumulate more readily in brains that are clearing less efficiently across the night. But here is where the story turns in on itself. The regions of the brain most essential for generating slow-wave sleep, the frontal cortex and the hippocampus, whose circuits must cooperate to produce the slow oscillations that drive deep NREM, are also among the regions where amyloid plaques accumulate earliest and most densely in Alzheimer's disease. So reduced slow-wave sleep allows more amyloid to accumulate, and accumulating amyloid damages the very circuits that would otherwise generate the slow-wave sleep that clears it. The two processes move toward each other across the years, each one making the other more likely, the circle drawn more tightly with each passing night of insufficient deep sleep. This is not a certainty about any individual life. It is a pattern that emerges from population data, from longitudinal studies following thousands of people over decades, from brains examined in laboratories and tissue banks. Large prospective studies, ones that followed participants over many years and watched what happened to their health as their sleep was measured and recorded, have found that sustained periods of short or disrupted sleep across the middle decades of life are associated with brain aging markers visible on magnetic resonance imaging, with higher rates of amyloid accumulation, and with elevated risk of dementia in later decades. The brains of people who slept poorly for many years show, in some studies, measurably more atrophy in the frontal and hippocampal regions, the same regions most vulnerable to amyloid deposition, the same regions most responsible for generating the deep sleep whose absence allowed the amyloid to accumulate. The structure of the relationship is not a cliff edge, but a slow incline, walked across many years, its gradient gentle enough that no single step feels consequential, but its destination, viewed from a distance, is far from where the journey began. Alongside all of this, there is still REM sleep. The older brain has not lost it entirely. REM sleep, that strange, almost paradoxical state in which the cortex fires with nearly waking intensity, while the body's muscles are held in stillness, in which the eyes move under their closed lids in quick, irregular sweeps, in which dreaming is most vivid and most likely to be remembered on waking, remains present across old age in ways that slow-wave sleep increasingly does not. Its percentage of total sleep time may decline somewhat, but it persists, and whatever function it serves, and there is more than one current understanding of its roles, none of them yet fully settled. Its persistence in older sleep is one of the features that marks the aging sleep architecture as genuinely reorganized rather than simply degraded. Different functions weigh differently at different ages. The balance shifts. What a sleeping older body experiences, moment to moment through a night, has its own particular texture. Sleep onset may take longer than it once did. The first deep NREM episode of the night may be shallower, shorter, or nearly absent. The first REM period, which in a younger adult arrives approximately 90 minutes into the night's first cycle, may come sooner, the pathway to it shortened now that the deep NREM that precedes it has thinned. The cycles that follow are lighter, brief arousals lasting seconds, too short to remember in the morning, but long enough to interrupt the slow electroencephalographic rhythm, occur more frequently, scattering across the night like small interruptions in a long sentence, each one brief, but each one adding to the accumulated shallowness of the record. By 4 or 5 in the morning, the internal clock has often already begun its daily rise, cortisol beginning its early ascent, core body temperature beginning its slow climb from its overnight trough, the biological morning arriving while the clock on the wall still reads an hour that would, in younger years, have held the body in deep sleep. There is something in this that has nothing to do with failure or disease. The body that wakes early in old age is not a broken body. It is a body whose clock has shifted, whose deep sleep machinery has changed its calibration across the years, whose architecture has reorganized itself around the demands and resources of a later season of life. The neuroscience does not tell a story of decline so much as it tells a story of transformation, a real transformation with real costs and real consequences, but one that has been running along the same biological lines in human bodies for as long as there have been human bodies old enough to experience it. Every person who has lived long enough has passed through these changes. Every elder who has lain awake in the early morning hours while the world outside still sleeps has been following a pattern laid down not in individual failure, but in the biology of the aging clock. Still, the costs are not nothing. The research is careful and the data are consistent. The quality and quantity of sleep across the adult lifespan accumulate into a long record, whose effects are legible in the brain and body. Sleep is not a nightly reset that returns the system to exactly the same baseline it left. Each night's sleep does what it can with what it has. When the slow-wave machinery has been running at reduced efficiency for years, the overnight clearance of metabolic waste is somewhat less complete. When the slow oscillation and the sleep spindle have been drifting slightly out of synchrony for decades, the nightly transfer of certain memories from short-term to long-term storage is somewhat less efficient. When the circadian signal has been growing softer with each passing year, the timing of all the systems it coordinates has been growing gradually less precise. The aggregate of these changes is not felt in any single night. It accumulates the way sediment accumulates, each night adding its thin layer to what came before, the record growing slowly denser, its weight increasing across the years without any single moment that could be named as the turning point. And yet, none of this is the final word, because the science does not end with a reckoning. It ends, if it can be said to end anywhere, with the observation that the system is still running. In a 75-year-old body, lying still in the dark, the suprachiasmatic nucleus is still cycling through its molecular rhythms, its clock genes turning on and off in their ancient coordinated sequence, still generating the 24-hour oscillation that organizes the day. The remaining slow-wave sleep, however brief, still carries its clearance function, still drives whatever glymphatic flow is possible through tissue that has reorganized itself around decades of use. The sleep spindles that remain are still gating the brain against unwanted intrusion, still participating in whatever memory consolidation the night can accomplish. The REM sleep that persists still serves its purposes, whatever they are, in whatever proportions they apply at this age, in the long dreaming darkness between midnight and first light. The system has changed. It has not stopped. In the fluid-filled channels that run along the blood vessels of a sleeping brain, the cerebrospinal fluid is still moving. It moves more slowly than it once did, through a network that has changed across the decades, in response to slow oscillations that are less frequent and less deep. But it moves. The amyloid beta molecules and the tau proteins and the other metabolic residue of a day's worth of neural activity are still being carried outward from the interstitial space, still being swept along the perivascular pathways toward the larger drainage channels that will carry them away from the brain. The tide is lower than it was in young adulthood. The clearing is less complete. And still, the tide moves, because the slow oscillations that drive it are still present, fainter, rarer, but real, and the channels that carry the fluid are still there, as they have been since the brain first formed, as they will be until the last night of a life that is still, tonight, not over. This is what the long view of sleep looks like when seen across the full arc of a life. It begins in an unformed brain, cycling through states it cannot yet organize into the clean architecture of adult sleep, its cycles short and its stages blurred, its circadian clock not yet calibrated to the turning world. It deepens through childhood into the richest slow-wave sleep that the human brain will ever generate. Enormous delta waves rolling across the cortex in the first hours of the night, their amplitude greater than at any other age, their homeostatic function running at its most powerful. It reorganizes through adolescence as the circadian clock shifts its phase, and the frontal cortex begins its long remodeling, the slow-wave sleep of childhood giving way to a later, different architecture. It persists through adulthood in gradually changing form, its deep stages slowly thinning, its efficiency slowly decreasing, its coordinating rhythms growing slightly less precisely, coupled with each passing decade. And it arrives in old age at a new configuration, lighter, earlier, more easily broken, still organized around the same fundamental mechanisms that were present on the first night of life, but carrying the evidence of every year that has passed since then. The slow oscillation that moves across the sleeping cortex is, in a sense, the oldest rhythm in this story. It is the wave that defines deep and REM sleep. It is the oscillation that drives the glymphatic tide. It is the rhythm whose power peaks in early childhood and diminishes through every subsequent decade. It is the frequency, less than 4 cycles per second, less than half a cycle per second at its most characteristic, that sits at the lowest end of the brain's electrical repertoire, the deepest known oscillation that the sleeping cortex generates. In a sleeping 5-year-old, this wave sweeps through the frontal cortex with an amplitude that no subsequent period of life will match. In a sleeping 70-year-old, it still appears rarer, smaller, briefer, but its electrical signature is recognizably the same wave that moved through the same kind of brain 7 decades earlier. It has not changed its nature, it has changed its scale. What the science of sleep across the lifespan ultimately reveals is something that no single age can show alone. It is the shape of the whole arc, the way a biological system built for one set of demands in infancy reorganizes itself continuously across 6, 7, 8 decades, adapting to the changing requirements of a brain and body that are themselves perpetually in transition. The architecture of sleep is not a static structure that the body maintains across a lifetime and eventually loses. It is a living architecture reshaping itself in response to the developmental demands of each stage, the hormonal changes of each transition, the accumulated wear and maintenance of each passing year. Sleep in old age is not what sleep was in middle age. Sleep in middle age was not what sleep was at 20. Sleep at 20 was not what sleep was at 6. And sleep at 6 was not what sleep was at 6 weeks, when the brain was still building itself in the long dark of the night. Every one of these configurations was the right configuration for its time. Every one was a response to the specific biological situation of the brain and body at that moment in the lifespan. The slow wave richness of early childhood was not excessive. It was exactly what a brain undergoing massive synaptic construction required. The phase-delayed circadian timing of adolescence was not defiance. It was the biological expression of a maturing pacemaker operating on its own reorganizing schedule. The declining spindle density and slowing delta power of midlife were not failures. They were the natural trajectory of circuits that have been running for decades and are changing their character as the years accumulate. And the fragmented, phase-advanced, shallow sleep of old age is not a loss of sleep. It is sleep as it has always been, doing what it can in the brain it has. There are still many things not fully understood about this arc. The precise mechanisms by which the glymphatic system is regulated during sleep are still being worked out. The full set of functions served by REM sleep across different ages is still a matter of ongoing investigation. The degree to which the sleep changes of aging are inevitable versus modifiable, how much is written into the biology of the aging brain, and how much depends on health, environment, and the accumulated habits of a lifetime, is a question that large prospective studies are still in the process of answering. The science here is not finished. It is a living body of knowledge, still being added to each year, still discovering things that change the shape of what was previously understood. The honest position, looking at all of it, is not certainty. It is the recognition that the broad outlines are clear, and the details are still being filled in, and there is no reason to expect that the picture will simplify as the investigation continues. In the laboratory, in the early hours of the morning, a brain is sleeping. The age of the person it belongs to is not visible from the outside. Only the EEG record tells the story of what age has done to the architecture inside. The trace moves slowly across the screen. There is no great sweeping delta wave of childhood's depth. There are occasional slow oscillations, smaller, less frequent, but real. The homeostatic machinery still running, still generating what slow wave sleep it can. There are sleep spindles, fewer than there were decades ago, but present, the thalamocortical circuits still producing their brief rhythmic bursts against the quieter background. There is REM sleep, its characteristic low-amplitude mixed-frequency trace, unmistakable, the eyes beneath the closed lids moving in their quiet, irregular sweeps. and there is the slow, steady respiration of a body at rest, the heart beating its long, familiar rhythm, the cerebrospinal fluid moving in its perivascular channels through the expanding interstitial space of NREM sleep, carrying outward what the day has left behind. Outside, the sky is still dark. The street is quiet. The world that will come in a few hours, with its light and noise and demand, has not arrived yet. For now there is only the sleeping brain, its ancient rhythms running at their aged pace, and the fluid moving silently through the channels of the tissue, and the slow, incomplete clearing that the night still manages to accomplish. The clock has not stopped. The tide has not withdrawn. The system that has been running since before the first breath of this life still runs, patient and biological, its mechanisms changed by time, but its purpose the same as it has always been, to hold the brain in the darkness for long enough to do what only the darkness allows. The cerebrospinal fluid moves along the vessels. The perivascular channels open. The interstitial space widens in the long pause between one slow oscillation and the next. The waste proteins drift outward through the expanded space, caught by the moving fluid, carried away from the neurons that made them, swept along channels too fine to see, toward the broader pathways that will carry them from the brain. This has been happening in human brains for as long as there have been human brains. It happens in infancy, in childhood, in the sleeping decades of adulthood, and it happens here, in old age, in this particular brain, on this particular night, in the quiet dark hours before the world begins again. The slow oscillation moves across the cortex, less than one full cycle per second, a long rolling wave, its peak and trough separated by more than half a second of electrical quiet.